So, I'm surprised it took somebody so long to figure out that we hadn't had a joke yet. So here we go. Ready? We have 2 today. You have to think about the range of the authors in age. Now that's a disclaimer. Why do giraffes have long necks? Because their feet are smelly. Of course. How about this one? How do you have a successful solar system party? Planet. Nicely done. You had one, haven't you? Yeah, you were right on that, man. You knew exactly what the answer was. Nicely done. Okay. What's that? Do you have one for me? I have a lot more, but we have to pace ourselves. We can't be overly hilarious. Okay. All right, so this is our last lecture. What's that? I didn't hear it. Yes. Last lecture, you didn't let me finish before the exam. All right, so you guys really should be working on some homework and understanding structurally where these features are located. Okay, this would be sort of objectives for this component is if you can draw a sarcomere and you can label these parts, you're probably doing pretty well architecturally or structurally, anatomically. That's another way of saying it. Remember, in this class, the structure or the anatomy is going to give rise or help facilitate the function. So we're talking about a very specialized cell, and these cells are arranged in a very specific order because they carry out a very specific function. And so understanding the architecture or the anatomy is critically important for being able to determine the function. So here is an answer key for the homework that I'd like you to do. Okay, are you going to turn it in? Well, you can consider Wednesday the day you have to turn in everything. So yeah, do it. It is for points, and you're going to be asked about it on Wednesday. Okay? Yeah, take pictures. Right? You want me to stand up there and pose next to it? Or?
Yes.
You actually have these slides, just in case you're wondering. You don't necessarily need to take a photo of it. I mean, they're actually on BB Learn. Okay. Let's try this question. The specific part of the sarcomere structure that's only occupied by thick filaments and is lighter in color than the rest of that particular band is called what? The H zone. B is the answer. Very good. Okay, so some examples of types of questions that you'll see on Wednesday, right? Talk about excitation-contraction coupling. Well, we already did. We're just recapitulating it. We're saying it again. But we're going to say it again in a different way. We have really 4 phases that you could help categorize in your head in preparation for Wednesday's exam. We have excitation. This is the process by which the nerve action potentials create muscle action potentials. Excitation-contraction coupling. Those are the events that link the action potentials on the sarcolemma to the myofilaments inside. And what's the key molecule that's responsible for that? Calcium. Contraction. That's the step where the muscle fiber develops tension and it shortens. Relaxation. This is when it's all finished and you want to relax and return to its resting length. So these pictures should look extremely familiar because we legitimately just went over them. They're just bigger. Okay, like, ooh, that was really confusing. It was just, the blow up. So these are the same pictures that we saw, but they're larger and there's fewer per slide. So, number 1, we get arrival of the nerve signal at the end of the axon. the details of the neuromuscular junction we went over in a previous lecture at detailed length, right? And this nerve signal opens these voltage-gated calcium channels in the synaptic knob. That's the end of the axon. Calcium, you see the little white calcium floods in. As it floods in, it binds to receptors on the vesicles containing acetylcholine, and those are encouraged to go fuse with the membrane and dump acetylcholine into the cleft. So acetylcholine is now in the cleft. If we blow up this postsynaptic side, also known as what? Motor end plate, nicely done. You can see is this magnified view. You've got this purple-looking acetylcholine receptor that has two binding sites, two little green acetylcholine molecules bind to it and open up the channel. You tell me what happens next. It opens up the channel and what comes in? Sodium. Okay? Sodium comes in. So in higher magnification view, some of you are like, I can't quite see. Right? So that's this picture. This picture is just this one blown up in higher mag. So here you've got an acetylcholine binding. It's going to open this channel that's a transmembrane protein through the phospholipid bilayer. We call it a fancy term known as the sarcolemma in a muscle cell. Opens this channel and sodium rushes in. We've already gone over this in great detail because this is exactly the same way that we propagate action potentials down a nerve. Okay? The only differences that we see here is that the resting membrane potential of a muscle is at minus 90 instead of minus 70. It's just the way it is. Okay? Slightly more negative inside of a muscle. Likely because the contents inside the muscle, there's more protein in a muscle cell than there is in nerve. So in a nerve cell, there's fewer protein. Right? In a muscle cell, there's tons of protein, like troponin, actin, tropomyosin. Remember all those? So a lot of those proteins are negatively charged. That makes the inside of the cell a little bit more negative than a typical resting nerve at minus 70. Okay? But you can read this and say, okay, this makes sense. Resting membrane potential goes from minus 90 to positive 75. Then potassium exits, and it returns back to resting membrane potential. This quick switch here we call an end plate potential. So this action potential on the muscle interface is called an end plate potential. Remember we called it a motor end plate? So now we call it an end plate potential because that electrical signal is on that end plate. All right, we already saw this picture too. It was just really, really tiny. So I blew it up so it's easier to see. Because I know you guys print like 25 images per page. So you're like, with a Mac, you can't even tell what's on there. So hopefully you can see this. Here is our synaptic knob on the upper right. Down here is the membrane of the muscle, known as the sarcolemma, phospholipid bilayer. You can see the acetylcholine receptor here. As the sodium comes in and triggers an end plate potential, that moves along the membrane and triggers our voltage-regulated ion gates to open respectively, just like we saw with the nerves. Okay? So the last unit on the nerve action potential, it's the same mechanics that we see here. Another example of how things are extremely cumulative.
So the ligand-gated channel comes first, and if enough sodium is let in, then the voltage gates open?
That's correct. Yep. So first this guy opens. If sodium passes threshold, if the sodium influx is enough to cross threshold, then you actually trigger these guys. Okay? All right, so now, if we look at where we are in this membrane, we zoom in, we're right here. Do you see these three little arrows? This third one kind of dives down. So we're right at this interface. And now we need to do excitation-contraction coupling. So we need to get calcium involved. You guys with me? So when this signal comes down, this specialized structure known as our T-tubule, which is an extension of the sarcolemma, the membrane of the muscle cell, it allows for that electrical signal to get into the interior of the muscle. Because the interior of the muscle cell is where the inside mechanics of thick and thin filaments reside. And unless you can get that electrical signal into the depths of the cell, you're not going to get adequate contraction. Okay? So this signal rushes down the T-tubule. Conveniently located on either side are these nice large containers of calcium, known as what? Terminal cisternae. And they are part of what larger network inside the cell? Sarcoplasmic reticulum. And the function of the SR and the terminal cisternae, which is a part of the SR, is to do what? Store calcium. So pretend that your automobile, let's pretend it's a small automobile. Okay? I have lots of children, so my automobile analogy is not a good one. Your automobile analogy as a college student is probably more appropriate. Let's say you have a two-door, sporty-looking car. Okay? The amount of calcium that you can pack into the car while people are occupying the seats in the car, let's pretend your trunk is empty. So calcium is going to be packed in your trunk space and in the inside of the car. The inside, the whole car is the sarcoplasmic reticulum. The trunk is the terminal cisternae. Okay? You guys follow that analogy? All right. So now we have this signal here that's in close proximity to our terminal cisternae. Conveniently, this opens voltage-gated ion channels in the T-tubules as a result of that electrical signal coming down. Those gates open calcium channels that allow calcium to enter the cytosol of the muscle. Okay? So now you've just triggered calcium influx into the inside of the cell. It's kind of hard to imagine because you're storing your calcium stores in a structure that's located inside the cell. Because the sarcoplasmic reticulum and the terminal cisternae are components inside the architecture of the cell. But if it's inside of that structure, it's not freely floating in the cytosol to actually interact. You can see now how it's flooding out. Now it can actually interact with troponin. So let's look at this picture on the left first. Here we've got troponin in yellow. We've got our white calcium molecule binding. And you can see that the troponin is physically connected to tropomyosin. And that tropomyosin is a long, slender-like protein, filamentous, and it physically covers over a binding site. And so when calcium binds, and this changes the architecture of troponin, it tugs on tropomyosin, and you can see, going over to this one, now tropomyosin slides out of the way, exposing the binding sites for the myosin head on our actin molecule, which is the reddish colored globular proteins. Now once that happens, this myosin head can actually bind and form what? What would we call that? A cross bridge. We call that a cross bridge. Okay? You guys are starting to get it. That's good. Okay, so now we're going to go through contraction events one step at a time. We're going to go through the cycle. Well, when we start out, the myosin head is in what we call sort of a cocked position. It's in a ready set position. And when it's in this position, it's actually already cleaved off one of the phosphates. Not all of it, just one of the phosphates. It still kind of hangs around, but cleaving that phosphate is what actually allows it to cock back and be ready, because now it can actually see and be in the right orientation to bind to the binding site. Okay? The myosin head has an enzyme, an ATPase enzyme, located within the head. That's actually why it's kind of bulbous, because it's housing the enzyme. Otherwise, it would just be a thin strip. But it's a thin strip, like a golf club, and then it has like this bulbous head. Did you guys watch the Masters this weekend?
Yep.
No? Did you guys know that the winner is like your age?
Yep.
Isn't that awesome? Yeah. And they kept saying, if he would have stuck around for another month, he would have graduated. And then the announcer's like, who cares, right? It's awesome. It's like national TV, and they just like totally trash the educational system. But the guy did win the Masters, and he's got some skills. So unless your golf game is that good, you should probably really focus on Wednesday. Okay. What was I talking about? Oh, golf clubs. Okay. So this bulbous head houses this myosin ATPase. Or I'm sorry, yeah, it has a myosin ATPase in the myosin head. Now, when it's cocked, it's activated, it binds, it interacts, here's our cross bridge. Okay, now we move forward. And the power stroke is, the cross bridge is formed, and now it strokes forward from the cocked position, it strokes forward. It's like throwing a ball, right? When you're going to throw a ball, you take the arm back, and then you throw it forward. And that's exactly what happens with this power stroke. Okay? Now you have used up all of your energy that's bound to that myosin head in that power stroke. And you're still physically attached. So in order to release, you actually have to bind another new ATP molecule. A new ATP molecule has to bind. And this process repeats itself in the video as many times as you saw that, cocking in action. Every time it cycles, that's one ATP molecule that's being burned. So if you go back and you watch the video again, you'll actually see the artist's representation of ATP cycling really, really quickly. Okay, so as you wave to your friend, you have ATP cycling every time you're contracting. Okay, so think about all the muscle activity we do throughout the day just to stay alive. Respirations, cardiac cycle, okay? Digesting your food. Smooth muscle, cardiac, and skeletal muscle all have cross bridges that form and use ATP. Cardiac and skeletal are organized very similarly, and smooth muscle is organized differently. But it still has actin and myosin. Okay, so this activity is about 5 power strokes per second. That's how quickly this takes place. In each power stroke is going to use one molecule of ATP. Okay? Well, we're not done yet with the cycle, and then I want to show you another drawing when we're done. Relaxation. In order to relax, you have to bind a new ATP, but now you need to get rid of the acetylcholine. It's sending the signal it was asked about earlier. So you stop sending the nerve signal to start relaxing, and you also stop releasing acetylcholine, and you allow for acetylcholine esterase. You can see this purple moon-shaped structure chewing up acetylcholine that's left over in the cleft. The fragments are reabsorbed, and you stop sending your active signal. Now, in addition to that, remember the events that happen within the muscle itself. You have calcium that needs to be reabsorbed and get out of the cytosol. So you conveniently have a network structure that stores calcium inside the cell. It's very close by. It's very close by to those filaments. And so you pump it actively back into sarcoplasmic reticulum by active transport. That keyword active transport should tell you that it requires ATP in order to run that. Calcium binds to calsequestrin that's in the sarcoplasmic reticulum. Right? That analogy would be like in your trunk you have, you put your suitcase in and then you have like a bungee cord with like hooks on that so your suitcase doesn't fly around every time you turn. Because I know I've seen you guys drive. You need that. Okay? ATP is needed for relaxation as well as contraction. Okay? So calcium, in our relaxation summary, is removed from troponin and it's pumped back into the sarcoplasmic reticulum. Now, without calcium bound, right? Tropomyosin reblocks the sites. The sites get reblocked, and now myosin can't interact with actin anymore. And you stop producing tension, and the fiber itself returns to resting length. Because as it contracts and gets shortened, if they stop touching each other with tighten, the elastic filament, it actually naturally recoils back to its resting state.
Does something initiate calcium unbinding, or does it just release?
It just releases. Yeah, it has a light affinity for troponin. So troponin will bind it, but not nearly as aggressively as calsequestrin. Okay? Other questions? Okay. Never mind. Okay. Never mind. Question?
So I'm still kind of lost with the ATP usage. One ATP molecule binds to allow the head to attach to the active site, or the active site. Is that true?
It started from binding an ATP molecule. And if you go all the way around, you bind an ATP molecule, right? You cleave off one phosphate.
To talk back.
Right?
Okay.
And then you have ADP plus PI, right? That allows for the power stroke to take place. And now you've burnt everything up.
Okay.
You're done. Now everything's finished. In order to release and then immediately recock, you bind a new ATP molecule. Okay? All right. So here's what I want to do. I'm the world's worst drawer, but I think this will be helpful for you. Okay? You guys like my myosin head? I'm only doing this if you don't laugh at my artistic non-skills. Now, remember that this structure is full of these heads. Go back to the video. Okay? So this is our thick filament. Now, we have a thin filament that's nearby. Right? Our thin filament is made up of 3 proteins. What do you think this one is that I'm drawing? That's actin. Okay? So the red one is actin. You guys all read that? Okay. What's another one? I could call on you guys to come up here, but we don't have a lot of time. But I think drawing these things out is helpful. Okay, how about tropomyosin? Let's draw tropomyosin, and we're going to draw tropomyosin in this blue color. And tropomyosin is sort of like this long, do you remember this filamentous-looking protein? Yeah. Okay. Okay. Now, if I was to note on here with an X, and obviously this is on all of them, what are those X's that I'm noting? It's the binding site for myosin. Okay? Pretend, even though this one looks like it's exposed, we'll put this here. All of these are covered by tropomyosin. Okay? So the blue tropomyosin. Got it? Okay, we're missing one key protein. Troponin. Okay, so troponin, we're going to actually make it... What color did I decide? Ah, I decided green. Good call. Okay, troponin. What binds to troponin? Where does the calcium come from? That's not a poop. Oh. How about now? You're like, oh yeah, that does look like a poop color. That's funny, the other day my wife said that. I don't know what we were talking about. And my kindergartner's like, Mrs. Hayes says you can't say that word. Mrs. Hayes is her kindergarten teacher, so hats off to Mrs. Hayes. Thank you for teaching our children to not fall after mom and dad. Okay, so in the terminal cisternae, okay, terminal cisternae, we have calcium. right? Calcium gets released, and where does it go bind? To troponin. Okay? So it binds to troponin. Okay, this would be something you do in SI. Oh man, it's all wonky now. Hang on. My lighting is bad. How do I turn my light down? Brightness. Oh, look at that. Oh yeah. If I go really dark, then it looks way better. Okay. All right. Now calcium just bound to troponin. So what's going to happen? Okay, so now we're going to get this movement that takes place, right? We actually need to sort of draw a second picture. But the picture that we need to draw here, we could actually just blow up and we could do this. Right? Those are our binding sites. Where's my blue? You with me? What's the brown one? Calcium. Okay? And I'll draw it like this. Okay. Does that make sense? So that's like the third time we've explained that today. Just today. Right? Next up, I'm going to sing a song with a harmonica. Yeah, that would be rad, but I would never be able to do that. Yeah.
Would it be wrong to say calcium stored?
Say that one more time. What about?
Would it be wrong to say calcium stored in the sarcoplasm?
No, that would not be incorrect. The question was, would it be wrong to say that calcium is stored in the sarcoplasmic reticulum? No. That is totally okay, because this structure is part of the SR. Okay? That confuses lots of students. That's why it's good to have these sort of Q&As, if you will. All right, is that helpful?
Yeah.
Okay, sweet. All right, so let's go back and talk about, in the remaining time that we have, some of the dynamics. Some of the dynamics of muscle. Dynamics of muscle. Well, if we look at this architectural pattern of the muscle, you can sort of appreciate there's three different states that are shown here graphically. You can see this resting length. And the way that you would measure that is you'd measure the distance from a Z line or disc to a Z line or disc, which is also referred to as what? Sarcomere. And the sarcomere is, by definition, the functional unit of muscle. Okay? So you can see the length here is about 2 microns, 2 to 2.25 microns in length. And that's showing up right here. This is a graph here of tension or force in comparison to length. So the idea is you get the most amount of force. You see how there's the highest amount of force here? When you optimize the overlap of the number of cross bridges that form. That should make sense. If you increase the number of attachments, the number of oars in the water, or rowers, you're going to generate more force. If you overly stretch the sarcomere out, then you can see that certain myosin heads don't even have the opportunity to find a binding site. They're too far away. Force goes down, right? Lower force on this side of the curve versus right here, higher force. Well, the similar relationship is found if you overcrowd. So if you contract it too much, you start losing force production Because now as you overcramp them, you get the lack of efficiency because you have maybe some more binding sites that are available, but the binding sites that are available right here in the middle, there's no myosin heads architecturally to find them. Okay? So I really like this picture because it shows this, what we call a length-tension relationship, and that there's a maximum amount of overlap that optimizes our ability to force, or produce force. Now, the central nervous system stimulates what we call a muscle tone, or a tonus response. It maintains a constant response of your muscle. There is a basal amount of tone in your muscle that happens even when you're not contracting it. So evidence of that is in stroke patients. So if a patient that has a stroke, you will see that basal tone of the facial muscles go away if they survive a stroke. If it's a unilateral stroke, then the one side of the face that isn't receiving nerve innervation is not receiving this tonus response. And so you get sort of this drooping of that side of the face. Okay? We also have what we call a cross-sectional area relationship. And this is saying that the greater the cross-sectional area of the muscle, the greater the amount of force that can be produced. There's no graph for this, you just need to write that in the bottom left, white space. So the larger the diameter of the muscle fiber, the more cross bridges that can form. Because the larger the muscle fiber, the more myosin heads are in that muscle cell, and so you can generate more force. So a larger muscle generates more force than a smaller muscle. That should make sense. And it's a direct relationship. Okay? As size goes up, force production goes up. Okay, so how about this concept? Why, then, do muscles contract at varying forces? Varying forces, or varying strengths is another way to look at it. So on the left side of this slide, what we're showing is some details around a muscle twitch. Okay? A twitch is a single firing of an action potential, or a stimulus. So you're telling the nerve sending a signal to the muscle, one signal saying, fire. That would be considered a muscle twitch. And you could measure it in electrical signal, otherwise known as voltage. And that's how we measure it. So you can see right here, here's threshold. And it isn't until about stimulus 4, 1, 2, and 3 don't cross threshold. Stimulus 4 crosses threshold. Right? I guess they're saying that stimulus 3 crosses threshold. So 3 on up, you get threshold reached. If you look down here, the individual nerve fibers that are being recruited are directly proportional to the strength of the signal. So the larger the voltage signal that goes, the more of the nerve fibers that you're going to recruit. So for example, it might be difficult to see the middle part, but if you look at this one, it looks more red. If you look at this one, it looks non-red. Okay? So it increases the amount of red intensity till you get to the far right based upon the number of individual fibers that are being recruited. The other thing that you can see is the number of fibers that are recruited, the electrical signal, is directly proportional to the response of the muscle and to the force or the tension that's being generated. So for example, if you recruit more muscle fibers, more nerve fibers, you get a maximum contraction, versus if you don't recruit as many, like here, number 3, it actually reached threshold. So 1 and 2 never reached threshold, you never fired the muscle. 3 reached threshold, but barely, so you get a small little muscle twitch. So the strength of the electrical signal translates to the strength of the intensity of the contraction. Okay? Now, how does this relate to additional things that we're going to do? Well, throughout our daily life, it's not just about changing the amount or the intensity of the nerve stimulation. You actually may need to recruit many, many motor units. So for example, in our twitch example, this is a different way of representing this. Single twitches, single muscle twitches. Well, you can also, over time, this would be an example of temporal summation because you're sending different individual nerve fibers. These are examples on the right of temporal summation. So spatial summation on the left, temporal summation on these 4 panels on the right. Spatial summation here because you're using the recruitment of different nerve fibers in different places. Temporal because you're summating them over different time sequences. So for example, if you compare this twitch on the upper left to the treppe response on the upper right. Treppe is a word that means staircase, a staircase phenomenon. So you can see the graph kind of climbs a stair. And what happens is, if you fire the second twitch before the first one comes all the way down to rest, you can actually increase the staircase. Okay? Or, excuse me, not before it comes down to rest. You fire it, it comes down to rest, and you fire it quickly right afterwards. It comes down to rest, fire it quickly right afterwards. But what happens is, this staircase phenomenon, even though it goes down to the resting state, it continues to increase in intensity because of the frequency or the closeness of sending the signal. Okay? Treppe, staircase phenomenon. Well, if you fire this fast enough and too close together, you get what we call incomplete tetanus. Incomplete tetanus is the staircase on the top with the staircase phenomenon on the bottom, meaning you haven't given it enough time to come down to rest before you fire it again. So you can kind of appreciate where, as you're halfway here, you're not resting that signal before you fire the next one. You can also see the closeness here versus how much closer these guys are. So this is an additional way to recruit. An additional way to recruit where you spatially fire, and then, or temporally you fire, and before the time allows it to come back to rest, you fire again. And now you can increase the rate of, or the force production that happens in that muscle. Okay? So, an example would be like holding a heavy object in your hand, like your backpack. So take your backpack and hold it out like this. Eventually it starts getting a little harder to hold, right? And then you start getting tired and fatiguing. And then you start doing this number. You're trying to recruit additional muscles. Okay, so when that happens, you're going from here to here. Then you move to here. Eventually, with complete tetanus, is when you can no longer maintain it and the backpack falls. Okay? So any type of postural muscle, any type of sustained contraction would follow this kind of sequence from here to here to here to here. Okay? And that's how we recruit different levels of strength within our muscles. Now, we can characterize the different types of contractions based upon whether we see movement or no movement. So, for example, a contraction is force plus a length. So if we have an isometric contraction, which is shown on the far lower left, isometric, you generate tension or force without changing length. So that's like holding the backpack. Here it's holding a dumbbell out. Okay? If you have an isotonic contraction, you're going to have an isotonic in one of two different variations, the middle picture or the far right. You can either have a concentric contraction, which is you generate force while the muscle shortens. So that would be like the upswing of a bicep curl. Okay? You can see the arrow showing the movement going this way. An isotonic eccentric contraction is why you generate force while you lengthen the muscle. That's like doing negatives, if you will. So as you lower the dumbbell, you're still generating force in the bicep, to control the descent. So instead of just dropping it, you actually control the descent, and that's considered an eccentric muscle contraction. So most of the time in the gym, we'll look at rotating between eccentric and concentric contractions to get different types of force generation in our training regime. Okay. Now, I would be not doing us any favors if I didn't explain where this energy comes from. Because as we contract our muscles, we have it pretty dialed in that we need ATP. Where do we get our ATP from? Okay, we have a couple of different sources. This is all review, but now in the context of muscle, it's a little bit different. How so? Well, it's different with respect to how you recruit the energy. So this isn't different. This slide is basically saying, I can get ATP from two main sources. Aerobically, at the bottom, or anaerobically, at the top. So aerobic respiration, right, this is going to be slower. It's going to require oxygen. It's going to need to go through glycolysis first, but then shuttle into the Krebs cycle, and then into the electron transport chain. So the benefits is that you generate much more ATP in the presence of oxygen. That's aerobic. The negative is that it requires oxygen, and it's actually slower. It takes a little time for aerobic respiration to kick in. Okay? In an activity, about 40 seconds. So anything that you need to do between now and 40 seconds from now is not using aerobic respiration. Okay? Like, oh dude, there's a bear coming. That was like 10 seconds. Still coming. Maybe he thinks I'm dead because I'm just standing there. That's 30 seconds. I know, this is part of the lecture. I wanted to show you how long it takes. 5 seconds left. Now I can run. Okay? So how do you think that works for muscle, exercising muscle? We only had aerobic. Super great. Lots of energy. Not so great if a bear's about to eat you. Anaerobic. Doesn't require energy. It doesn't require energy. It doesn't require oxygen. So that's a benefit. Problems are, the negatives are, it doesn't make a whole lot of ATP. Now you got to figure out how to store it. It also has some kind of crummy types of side effects, like lactic acid. Okay? Well, if you look at our immediate sources, less than 10 seconds. Because these would be critically important for that bear situation that we just uncomfortably demonstrated. Okay? You've got phosphate donations that can come from two main sources. Right? So you can see here, here's adenosine diphosphate going to adenosine monophosphate, stripping off phosphate. That's what myokinase does. Myokinase is an enzyme that's found in the muscle. Myo, kind of referring to the muscle. It donates it to an ADP, adenosine diphosphate, and forms ATP. So you strip it off of one source and you give it to another. Well, you can also do this with creatine phosphate. Creatine kinase, also available in muscle, will strip a phosphate off of creatine, phosphate made creatine, and donate it to ADP and give you fast, quick ATP. And you've seen folks that are training use supplements of creatine phosphate. They kind of take it in their dietary intake. Okay? The reason is it gives you this quick energy in the first 10 seconds by donating phosphates. Okay? If we look at the first 10 seconds, right, we've got our phosphagen system. That's what we're talking about. We're stripping it either from myoglobin or from creatine phosphate. Okay? Then from like 10 up to about 40, right, we need a second location, and that's actually our glycogen system. That's glycolysis, anaerobic. Okay? That gives us 2 ATP net, right, per cycle of glucose molecule. But beyond 40 seconds, we actually need aerobic respiration. That's where it kicks in, is beyond 40 seconds. Okay? So that's where we get our ATP to do these kinds of activities. Questions? Okay, listen up. This is where we're going to pause. Up until here is game on for the exam. We will start here in the next unit, and then we will move on from there. Okay? See you guys Wednesday. Your bonus opportunities are in the back if you wanted to pick them up. And then SIs, there's a busy SI schedule tonight. Okay, so I would check PB Learn for those new posted times.